In this lecture, we will discuss about the biosynthesis and degradation pathways of proline. Proline is a glucogenic non-essential amino acid, that is, mammals can synthesize it by themselves, and essential amino acids are those which cannot be synthesized by mammals. Alpha amino nitrogen in the form of glutamate is an essential nutrient for non-essential amino acid biosynthesis. Proline is considered to be belonging to glutamate family. The other members include arginine and ornithine. Glutamate is the initial precursor for the synthesis of proline. So let us first learn about the synthesis of glutamate. Glutamate is synthesized by glutamate synthase, which is an enzyme which occurs only in microbes, plants and lower organisms. Glutamate is synthesized into glutamine is synthesized into glutamate with 5 steps involved in it. Initially, the electrons are transferred from NADH to FAD to yield FADH2 in site 1 beta subunit. Later, the electrons are transferred from FADH2 to FMN in site 2 alpha subunit. Glutamine is hydrolyzed to glutamate, alpha glutamate and ammonia. This ammonia is transferred to site 2 alpha subunit through a channel of 31 angstroms. Ammonia reacts with alpha ketoglutarate to yield alpha aminoglutarate. Alpha aminoglutarate is reduced by FMNH2 to glutamate. Glutamine is normally hydrolyzed via the nucleophilic attack of the cysteine 1 sulfhydryl group on the glutamine C-gamma atom to form a tetrahedral sulfonate group intermediate. First, we will see the biosynthetic pathway of proline. Conversion of glutamate to proline involves the reduction of gamma carboxyl group to an aldehyde, followed by the conversion of internal Schiff base, whose further reduction yields proline. Glutamate acts as the precursor for the synthesis of proline. Glutamate, in the presence of gamma glutamyl kinase and ATP is hydrolyzed to ADP, forms glutamate 5-phosphate, wherein the gamma carboxyl group is attached with the phosphate group. This is an unstable compound. In the presence of dehydrogenases, it forms glutamate 5-semialdehyde. Glutamate 5-semialdehyde cyclizes with the help of Schiff base by the formation of Schiff base. This cyclic compound is called delta prime pyrroline 5-carboxylate. Delta prime pyrroline 5-carboxylate, in the presence of pyrroline 5-carboxylate reductase, is reduced to proline. Here NADH gets oxidized to NAD+. Now we will discuss the degradation of proline. It is the exact opposite reverse steps of biosynthetic pathway. Proline, in the presence of proline oxidase, forms pyrroline 5-carboxylate. Pyrroline 5-carboxylate spontaneously gets linearized to glutamate 5-semialdehyde. Glutamate 5-semialdehyde, in the presence of glutamate 5-semialdehyde dehydrogenase, reduces NAD and forms glutamate. Glutamate, in the presence of glutamate dehydrogenase, forms alpha ketoglutarate, which is the intermediate product of TCA cycle. Thank you.